Lesson 1, Virus Detection and Isolation. We're going to focus on cell culture. This content is part of the module MBS 7117, Molecular Microbiotechnology. It's housed in the Department of Biomolecular Lab Technology and Molecular Biology, abbreviated as BLT. It's part of the School of Biosecurity, Biotechnology, and Laboratory Sciences, abbreviated as SBLS, in the College of Veterinary Medicine, Animal Resources, and Biosecurity, abbreviated as COVAB, in Makerere University. I'm your facilitator, Dr. Penny Nansamba. So back to lesson 1, virus detection and isolation. And this time we're going to focus on cell culture. We are continuing this series, and this time we are looking at the culture of viruses in cell lines. Previous series looked like the detection of viruses and the various methods used. However, now in series 2, we are looking at the culture of viruses in cell lines. Our contents for today are: number 1, the rationale for the culture of viruses in cells. Number 2, culture of viruses in different cell lines. And we are going to focus on the primary cell lines, diploid cell lines, and the continuous cell lines. Our learning objectives are 3 and 4. By the end of this section, you'll be able to describe virus culture in cell lines and highlight differences in the cell lines used for virus culture. The culture of viruses in cell lines. We'll start with an overview. The culture of viruses in cell lines is a basic and important method used to isolate and propagate viruses. The cells provide an environment for viruses that is both consistent and controlled. The picture shows a confluent monolayers of cells with viruses in blue adhering to these cells. The viruses are going to gain entrance into the cells and start replicating, bringing us to the phenomenon of culturing or passage of viruses in cell lines. So there are many different cell lines, and we are going to start with the primary cell lines. Let's have an overview. The origin. The primary cells are derived directly from animal tissues, such as the lung parenchyma, monkey or bovine kidneys, or tumors, to mention a few. What are the characteristics? They have a limited lifespan. You can only passage them 5 to 10 times before they undergo a crisis and die. They maintain the diploid chromosome number, which is normal, meaning that they are genetically stable. They retain morphology and function of the parent tissue. So if it's epithelial, they'll retain those functions. If it's the fibroblasts, they will retain those functions. And lastly, they are anchorage dependent. They must attach to a vessel for growth. Now let's look at the justification and uses of the primary cell lines. We shall start with the uses. They're used in studies requiring physiological relevant responses. These include drug toxicity studies, virus culture, and also vaccine development, to mention a few. What are the advantages of using the primary cell lines? They're broadly susceptible to many viruses because they are physiologically relevant, they have maintained most of the parent characteristics, and they retain the natural gene expression profiles. We're still continuing with the justification of the primary cell lines. We realize to every advantage there's also a disadvantage. So the disadvantages are such: they have a higher risk of contamination with endogenous viruses. That means when you harvest the primary cells from the host, you may be actually picking endogenous viruses of this host, which you're going to bring into your lab, and they may affect your studies or the subsequent use of these primary cell lines. Another disadvantage is that they're difficult to culture. They require complex environment conditions. There's batch variation. So the primary cells that you harvest from a host this week may be slightly different from the primary cells you harvest next week or next month, which may cause slight challenges to your experiments. And lastly, they present ethical concerns since they're obtained from animals or humans. Here we are illustrating how we set up a primary cell line. Let's start with number 1. We obtain our host, our animal, and harvest the organs or tissue. We grind these or mince them, probably using a pestle and mortar. Then in number 2, we obtain a clean conical flask and suspend our organs or tissue mix in collagenase enzyme. This is going to digest the tissue adhesions, setting our cells free. Then thirdly, our suspension is filtered, and then we can centrifuge it to pellet the cells at the bottom of our tube. Number 4, we wash the pellet a number of times. Number 5, we do differential centrifugation so that we can collect the more dense cells at the bottom and the less dense cells at the top. And that is our primary cells, which you can use to seed any vessel. It's time for self-reflection. What have we learned concerning the primary cell lines? And this time we're going to look at study question 3. Define the primary cell lines. Number 2, give any advantage of using primary cell lines. Number 3, outline any ethical concerns that are resulting from the use of the primary cell lines. If you have any difficulty in obtaining the answers to these questions, please review the previous section. We're going to go to the next cell line that's used to isolate viruses, and this is the diploid cell line. Let's start with an overview. The origin: they obtained from fetal tissue such as human embryonic lung, and we have the MRC5 cell line, or they can be obtained from purified chick embryos, giving us the WI38 series. Now, these were isolated from the White Star Institute in 1961. The characteristics of the diploid cell line: they retain the diploid number of chromosomes, which is 46, meaning that they're stable. They have a limited lifespan, but they can be used for up to 50 divisions, which is slightly longer than the primary cell lines. They have contact inhibition, meaning the cells will stop dividing, migrating, or growing when they physically touch each other, and this enables a monocell layer. They are not transformed, so they are not cancerous, giving us the natural physiological conditions. We are still on the diploid cell line, and now we are going to justify their use. So they're used in vaccine production, and they have the following advantages. They are genetically and phenotypically closer to normal human cells than the continuous cell lines. So they would give us a natural environment, simulating a natural environment. Secondly, they have a lower risk of tumorigenicity, again giving us a natural environment. However, once we've got advantages, there are also disadvantages. They are more difficult to maintain compared to the continuous cell lines, and there will always be ethical issues due to the fact that we are obtaining them from fetal tissue or from animals. Now let's reflect on what we've learned on the diploid cell lines. We'll study question 4. Part 1, define the diploid cell lines. Part 2, give any advantage of using the diploid cell lines. Part 3, outline any concerns resulting from the use of the diploid cell lines. If you have any problems or any difficulties in answering these questions, please review the previous section. We're going to go to our third cell line, which is the continuous cell line. We'll start with an overview. These are immortalized cells. For example, the lahella, the viro, the Chinese hamster ovary or CHO cell line, the bovine hamster kidney batch 21 cell line, or the BHK/21 cell line. Let's look at the characteristics. 1, they have an unlimited span, so you can continue to proliferate them almost indefinitely. Secondly, they possess genetic and epigenetic alterations that enable them to evade normal cellular sequence and continuously divide. Thirdly, they have an abnormal number of chromosomes. We're still continuing with the characteristics of the continuous cell lines, and they are fast growing compared to the primary and diploid cell lines. They may grow in suspension without the need for a substrate, which was not the case in the primary and diploid cell lines, which are anchorage dependent. And lastly, they have lost contact inhibition and can often pile up, which is not really a good characteristic. So let's look at the production of the continuous cell lines. Where do we obtain them? You can get them from spontaneous transformation. So primary cell lines may spontaneously acquire genetic mutations during subculture, as we see with the lahella cells, which were obtained from cervical cancer. Then we can induce immortalization. So virus infections can cause cancerous growth. For example, the HEK 293 cells were transformed using the adenoviruses. We can use chemicals to induce the genetic changes in the cells, or we can also use genetic engineering to induce this immortalization, such as the case of the cancerous genes or expression of the HTER2 T genes in various cell lines. Or we can derive them from tumor tissues or cancerous tissues. So we can derive them directly from a cancerous tissue, such as the MCF version 7 cell line that has been got from breast cancer. Let's justify their use. So they're used in monoclonal antibody production and recombinant protein expression. What are the advantages? They're easy to maintain. We've seen that we can subculture them on continuously for almost an indefinite period of time. They have long-term availability with consistency, so there's no batch variability, and it makes reproducibility a feature that we can depend on, and this makes them suitable for use in research. Thirdly, this is possible to scale them to be used at an industrial level. But each advantage comes with a disadvantage. And we see that they lose the original tissue characteristics. Therefore, they do not present the normal physiological response. And the second disadvantage, that there is genetic instability. So there will be genetic drift over time, and the batch that you started with at passage 1 may not be the same batch of cells you have at passage 50. Now let's reflect on what we've learned using the continuous cells, and we shall look at study question 5. Part 1. Define the continuous cell lines. Part 2. Give any two advantages of using the continuous cell lines. Part 3. Give any two disadvantages of using the continuous cell lines. If you have any problems answering these questions, please refer to the previous section. We're still continuing with the self-reflection on the continuous cell lines, and this time we're going to compare them to the primary cell lines. So question 6 is compare the primary and continuous cell lines. Let's have a hint. Look at the origin, lifespan, chromosome stability, ease of maintenance, applications, growth rate, contact inhibition, carriage dependence, or genetic stability. And this would be typically a take-home exercise. So let's continue our lecture. And we're going to look at the steps involved in virus culture on the cell lines. We've identified our different cell lines that we probably wish to use. And now we're going to illustrate how we passage viruses on these cell lines. Number 1, we're going to start with an inoculation. We obtain a confluent monolayer of susceptible cells, and we add virus to them. Then this is followed by the virus adsorbing onto the cells. And we've tried to illustrate this with the diagram here. The virus binds to specific receptors on the host surface cells. And this is going to permit virus to enter the cells. We'll continue with our illustration of virus passage in cell lines. So after the viruses have gained entrance into the cells, we go to incubation. The cells will be incubated under optimum conditions, which is 35 to 37 degrees Celsius, using 5% carbon dioxide in an incubator for about 3 days or 1 week or 3 weeks, depending on how long it takes for the virus to grow. Then part 4, we're going to observe for the cytopathic effect, or the visible effects of virus growth which are evident in the cells. And once we see this, we can now proceed in part 5 to harvest the virus. So we're still looking at the steps in virus culture in cell lines, and now we are observing the effect of multiplication of the viruses on the culture cells, and this is the cytopathic effect, or CPE. So number 1, typically we are going to see rounding of cells coupled with the detachment or loss of adherence from the flask. So in this picture, we see a confluent monolayer. In the next one, we see cytopathic effect. The cells have rounded up and they have detached from the vessel or the flask. And this was seen when we were propagating the feline infectious peritonitis virus. So a second type of CPE that we may observe would be the cells forming syncytia or becoming fused together, as is seen in this diagram. We can also see shrinkage of cells, this is not shown, increased refractivity, also not shown in the pictures. We can see aggregation or clumping of cells, or we can see cell death or lysis. We're still continuing with the observation of multiplication of cells in cultured cells, the multiplication of viruses in cultured cells, and we're looking at an alternative method to observation of CPE, because not all cell culture where viruses are passaged will give you CPE. However, other signs apart from CPE that we may see include observing inclusion bodies. And in these diagrams, we are showing you the inclusion bodies, which are round organisms inside the cells. They may be depicted by the arrows, or you may see them around the circumference of the cells. This was in the case of rabies in a human being. We can also use heme adsorption tests to detect virus growth in cells, and it's depicted by that assay. And we can also use interference, where the growth of a specific virus in the cells will inhibit a second virus from growing in that particular culture, or we can detect nucleic acids. Now let's reflect on what we've just learned. The question 6 here is, how can we detect that virus is replicating in vitro in cell lines? Let's have a hint. We can look for CPE. We can look for the detection of antigen, nucleic acids, heme adsorption, interference, to mention just a few. If you have difficulty in describing what we've just discussed, please review this section. Now, in summary, the 3 different categories of cell lines, primary, diploid, and continuous, used in virus culture have been discussed in detail, considering the following themes: the origin and characteristics, merits and demerits of each, and we've also described the procedure of virus passage in these cell lines. Our next steps would be for you to refer to the lecture notes given to you on the study portal. the ETVT's chapters in the textbooks recommended to you for further reading also in the study portal. This has been our source of information. Thank you.